Welcome to The Explainer. Today we're on the case of a medical mystery, a real whodunit that unfolds inside millions of us every year. We're talking about the phantom stomach bug.
So, let's start with a situation that is just beyond frustrating for so many people. You get sick, you take the medicine, you get better, the tests say you're cured, and then, a little while later, it's back. How does that happen?
Okay, picture this, because it happens all the time. Someone gets diagnosed with H. pylori. That's a super common stomach bug. They do the whole course of antibiotics, they start feeling great, and a follow-up test comes back clean. Awesome. Problem solved, right?
Well, not so fast. A few weeks, maybe a couple of months go by, and that familiar, awful feeling starts to creep back in. They go back to the doctor, get tested again, and yep, H. pylori is back. So the obvious question is, did I just get unlucky and catch it all over again?
Here's the twist. Most of the time, the answer is no. It's not a brand new bug. It's the exact same one from before. It never actually left. It was just hiding, playing dead, with this incredible survival trick.
Yeah, scientists have a pretty cool name for this microscopic magic trick, the Viable But Non-Culturable state. Let's just call it VBNC. Think of it as a deep hibernation mode. The bacteria are totally alive, but they've shut down so completely that you can't grow them in a petri dish, which makes them basically invisible to our usual tests.
So how in the world does this tiny organism manage to activate its own personal cloaking device? It's a really amazing transformation. You see, H. pylori is kind of like a shapeshifter. When it's active and causing problems, it's this spiral shape, zipping around and full of energy. But when it goes into that VBNC state, it curls up into a tiny, dormant sphere. It slams the brakes on its metabolism, brings it down to almost zero, and totally stops trying to multiply.
And this is the part that is so crucial to understand. The bug isn't dying. This isn't a sign that it's weak or on its way out. No, this is a highly evolved, intentional defensive move. It's a survival program it runs to wait out the storm.
Okay, so it has this brilliant disguise, but how does it actually fool our best medical technology?
Well, these bacteria don't just decide to go into hiding for fun. It's triggered by stress. And what's the biggest stressor for a bacterium? You guessed it, antibiotics. But other things can do it too, like running out of food or big changes in the stomach's acidity. The bug basically senses danger and goes, nope, I'm out of here, and hunkers down.
And this brings us right back to the central mystery. If these little sleeper cells are still hanging out in the stomach lining, why do all of our gold standard tests come back saying the coast is clear?
This chart just lays it out perfectly. The problem is, our tests are built to find the active, spiral-shaped version of the bug. Stool tests, they look for proteins on the surface that the dormant bug just stops making. The breath test, it relies on a specific enzyme that the bug basically switches off when it goes to sleep. And taking a biopsy to grow it in the lab, that's a total non-starter because the very definition of VBNC is that it won't grow.
So, this bug is a master of hiding. But it can't stay hidden forever. So, what wakes it up? This is where the story gets really, really interesting. Waking up, what scientists call resuscitation, isn't like just flicking a switch. It's a whole process. First, the threat has to be gone. The antibiotics are out of the system. Then it needs a little help, like an antioxidant, to clean up some internal damage. After that, it has to literally rebuild its outer shell to get that spiral shape back. And only then can it fire up the engines and get back to business.
Now, you're not going to believe this, but one of the most powerful keys to this whole wake-up process is probably in your kitchen right now.
I'm not kidding. A really recent study, just from 2023, discovered that vitamin C is a potent trigger for waking these bacteria up. In the lab, they found that adding vitamin C could coax more than 60% of these dormant bacteria back to life. It's like it gives them the jumpstart they need to get going again.
The other major player in this is a protein with, the most perfect name ever, Resuscitation Promoting Factor, or RPF for short. You can think of it like a tiny mechanic that comes in and repairs the bug's outer wall, letting it stretch back out from a sphere into that infectious spiral.
So, finally understanding this whole amazing cycle, the hiding, the waiting, the waking up, is completely changing how we think about fighting not just this infection, but maybe a lot of others too.
So here's the bottom line. When H. pylori seems to come back, it's usually not a reinfection. The proper term is recrudescence, which is just a fancy way of saying the original infection reactivated. The bug that was already inside you just woke up.
And this isn't just trivia. It has huge, real-world implications. This is why doctors make you wait at least a month after treatment to test again. They're hoping to catch any bugs that might have woken up. It means a negative test isn't a 100% guarantee. But, and this is the exciting part, it opens up wild new ideas for treatment. What if we could use something like vitamin C to intentionally unmask the hidden bacteria, forcing them out of hiding so our antibiotics can finally wipe them out for good?
So that leaves us with this one last big question to chew on. If bacteria are this clever, if they've evolved these amazing strategies to literally play dead and hide from us, how do we ever know for sure that they're gone? It forces us to rethink what it even means to be cured.
Usually, bacterial infections are basically a game of whack-a-mole. You see them all, you hit it with an antibiotic, and the game is over.
Right, it's straightforward.
Exactly. But Helicobacter pylori, it unplugs the arcade machine and just hides under the floorboards. So today, we're critiquing a hospital study guide on this elusive H. pylori viable but non-culturable state.
And it's a fascinating piece of material, honestly.
It really is. Restructuring the presentation to lead with the clinical stakes rather than the molecular biology will dramatically increase engagement from your target audience of medical practitioners.
I completely agree, because right now the author is taking a massive risk with audience alignment.
Yeah, let's look at that opening section. The material starts with this incredibly deep dive into how the bacteria suppresses the TCA cycle.
It goes straight into the weeds.
Literally minute 1, it goes into detailing the activation of the upstars and POPGA genes, which, stabilize the coccoid shape and modify the peptidoglycan cell wall.
And the proteomics and transcriptomics presented there are undeniably rigorous. Like, the science is solid.
Oh, absolutely. But I'm putting myself in the shoes of a clinician who's sitting in a packed hospital ward. The weakness here is that presenting highly technical molecular data before establishing the clinical context risks overwhelming that clinical audience, causing them to completely disengage.
They'll just check out. I mean, if an infectious disease specialist is faced with slides full of downregulated enzymes like GLK or FBA right off the bat, they are going to mentally tap out before they ever understand the practical impact of this dormant state on their actual patient care.
Because the primary goal isn't to memorize the transcriptomics of the PGDA gene.
Right. Their goal is to figure out why their patients keep relapsing.
Exactly. So the suggestion is to invert the narrative structure by leading with the clinical mystery of eradication failure and false negatives, and then utilizing the molecular biology as the tool that solves that mystery.
Yeah, you turn the biology into the hero of a clinical detective story.
But looking at this transcriptomics data, I have to wonder if we're throwing the baby out with the bathwater by just telling the author to hide it. Like, how much of this genetic data actually needs to stay?
Oh, I think it all needs to stay. The science is the backbone of the entire guide. It's just about the sequencing.
So how would you actually restructure it?
Well, if I were doing it, I would probably put a highly frustrating, totally relatable clinical scenario right on slide 1.
Okay, give me an example.
So picture this. A patient undergoes standard eradication therapy for H. pylori. They seem completely cured. They stop their proton pump inhibitors, the PPIs.
Right, standard protocol.
Exactly. Then 4 weeks later, they return with a positive test and a massive resurgence of ulcers. And the clinician is just baffled because they used a proven, aggressive antibiotic regimen.
That is the exact visceral hook this piece needs, because once you've established that acute clinical frustration, then you can introduce the viable but non-culturable biofilm as a protective shelter.
Yes, you explain that the bacteria didn't suddenly mutate to become genetically resistant to the amoxicillin, it just went to sleep.
And that's where the genetics suddenly matter to the clinician.
Precisely. That is exactly when you bring in the UpPF's gene. You don't present it as just some abstract data point on a chart. You present it as the armor the bacteria use to fortify its cell wall and survive the barrage of antibiotics.
I love that framing. And then I guess you introduced the Micrococcus luteus-derived RPF enzyme, the resuscitation promoting factor.
The molecular alarm clock.
Yeah, the alarm clock. You explain that this enzyme acts as a muralytic agent, cleaving the cell wall and signaling the bacteria to wake up and resume its active spiral shape.
And when you frame it chronologically like that, the frustration, the hiding, the waking up, the transcriptomics feel like actionable intelligence rather than just textbook trivia.
It makes a huge difference. Grounding the advanced diagnostic methods in the everyday reality of hospital workflows will bridge the gap between cutting-edge research and clinical practicality.
That is a critical pivot. Because if we accept that the bacteria is hiding so well, we run into an immediate secondary problem.
Right. Once the clinician understands why it hides, the very next question they're going to ask is how they are supposed to actually find it. Which brings us to the entire section evaluating diagnostic methods.
And the draft is very focused on advanced lab techniques right now. The weakness is that positioning specialized, equipment-heavy tools like Viability PCR as the primary diagnostic solution ignores the acute resource constraints of standard hospitals.
Heavily focused on the advanced stuff. And, the underlying science makes total sense. Standard diagnostic methods fail spectacularly when H. pylori goes dormant.
They really do. The material explains that the stool antigen test yields massive false negatives because the bacteria downsizes its outer membrane proteins to avoid immune detection.
It's essentially pulling down the shades and turning off the lights. The antigens the stool test is looking for simply aren't there anymore.
And the urea breath test fails for a similar reason.
Because the urea breath test relies on the bacteria's active metabolism to produce carbon-13 carbon dioxide.
But earlier, the author noted that the bacteria suppresses its TCA cycle.
Exactly. It shuts down its main metabolic engine. So of course the breath test is negative. The engine just isn't running. So the material makes this incredibly strong case for alternative testing, specifically highlighting PMA-qPCR and flow cytometry with live dead staining.
Which is super cool technology.
Oh, I was blown away by the PMA-qPCR methodology, using propydium monoazide to cross-link the DNA of dead bacterial debris so that the PCR only amplifies the DNA of living, dormant bacteria. That is brilliant.
It's cutting edge.
But I have a major concern with this positioning. It's a bit like recommending a quantum computer to someone who just needs to balance their checkbook.
I see what you mean. The tools are undeniably powerful, but you're thinking about the logistics.
Yeah, I'm thinking about the community hospital doctor reading this guide. It potentially leaves clinicians frustrated and without a practical, immediate takeaway. How many everyday clinics actually have a flow cytometer or specialized viability PCR equipment ready for routine screening?
Almost none.
Right. If a doctor reads this, they're going to feel completely unsupported and helpless because they don't have access to the proposed solution.
We definitely don't want the reader feeling like the guide is entirely theoretical or out of reach. So the suggestion is to create a clear tier diagnostic triage pathway that distinctly separates routine clinical defense strategies from advanced academic or referral testing.
Like a visual decision tree slide, I think that would be incredibly highly utilized by the target audience.
Oh, absolutely. So step 1 of the tree should focus entirely on optimizing the timing of standard tests, strictly enforcing the rule of waiting 4 weeks post-treatment and 2 weeks post-PPI before retesting.
Let's trace the logic there. Since we know the dormant state causes false negatives on the urea breath tests and stool antigens, enforcing that waiting period makes sense. But I can anticipate a bit of pushback from a reader there.
How so?
Is that really a solution? It feels like we were just telling the doctor to wait and hope the bacteria wakes up enough to trigger a standard test.
It does require patience, yes, but there is a deep biological reason for it that the author needs to emphasize on that flowchart. You explain to the clinician why they are waiting.
Okay, unpack that a bit.
They are allowing the stomach environment to normalize. Stripping away the antibiotics and the acid-suppressing PPIs removes the environmental stress. And that normalization acts as a natural trigger for the bacteria to shed its coccoid armor, fire up its metabolic engine, and become detectable again.
Okay, that makes a lot of sense. It empowers the clinician to use time as an actual diagnostic tool.
Exactly. And then step 2 of the flowchart is where we reintroduce the advanced science.
Right. Step 2 should then reserve PMA-qPCR and live-dead staining exclusively for refractory, multi-drug resistant cases or academic research settings, framing them as specialized escalations rather than everyday necessities.
That bridges the gap perfectly. It gives the community doctor a practical, immediate takeaway they can implement tomorrow while still educating them on the horizon of advanced diagnostic referrals.
And accurately diagnosing a refractory case through that triage pathway naturally forces the clinician to confront the reality of how to treat a dormant infection.
Which leads us straight into the complexities of the proposed therapeutic interventions.
Framing the mixed clinical trial results of vitamin C as a deliberate, strategic protocol rather than a confusing contradiction will empower clinicians to utilize this adjunctive therapy confidently.
Because the data presented in the source material regarding vitamin C is incredibly dense. And frankly, it's confusing on a first read.
Extremely confusing. The weakness is that listing both positive and negative clinical trials without providing a definitive synthesized clinical stance leaves practitioners without clear guidance on how, when, or why to prescribe vitamin C.
Yeah, the material cites a highly detailed 2023 study by DiFermo and colleagues. In their in vitro models, when they induce the dormant state via nutrient starvation, adding just 1/4 of the minimum inhibitory concentration of vitamin C, which is about 512 milligrams per liter, resulted in a massive 60% resuscitation rate.
Wow.
Yeah, the bacteria's MTT metabolic activity spiked. It shifted rapidly from the dormant Coccoid form back to the active spiral form.
So in a petri dish, it is incredibly effective at waking the bacteria up.
Highly effective. But then the material pivots to the in vivo clinical trials, and they are completely all over the map.
Right. You have older studies showing massive eradication improvements, up to 78% success when vitamin C is added to bismuth therapies.
But then the author lists meta-analyses, like the one by Lee in 2011, or the El Shawi trial in 2020, showing absolutely zero statistical difference in eradication rates when vitamin C was added to standard clarithromycin triple therapy.
If I'm a clinician reading this and I see that a major 2020 meta-analysis essentially debunked it, I'm probably just going to abandon the vitamin C idea altogether due to the conflicting meta-analyses. I mean, why would I prescribe something that has such contradictory evidence?
That is exactly what will happen if the author leaves the section as is. You can't just tell a doctor, well, some studies say it works, some say it doesn't, but in a petri dish, it wakes the bacteria up.
Right. They don't know how or why to prescribe it to the actual human being sitting in their examination room.
So how do we synthesize this for the author so they can actually guide their audience? Because the conflict in the data is real.
The suggestion is to reframe vitamin C not as a standard antimicrobial treatment, but as a Trojan horse waking agent, paired with a highly specific, conditional prescribing protocol.
Let's unpack that Trojan horse idea. Why did the clinical trials with standard clarithromycin triple therapy fail so spectacularly?
Because standard antibiotics require active bacterial metabolism to work.
Exactly.
An antibiotic like amoxicillin targets active cell wall synthesis. If the bacteria is dormant, if it's not currently building a cell wall and is just sitting in its up-S-fortified Coccoid bunker, the antibiotic will literally just bounce off. It has no active mechanism to disrupt.
So the failed trials were essentially throwing weapons at a closed fortress. They didn't account for the waking phase.
Exactly. So design a conceptual slide that visually depicts vitamin C, drawing the Coccoid bacteria out of its dormant state so that standard antibiotics can effectively strike it. And to do that, they have to clearly explain the mechanism of ROS clearance.
The material mentions ROS, reactive oxygen species. Oxidative stress is one of the primary triggers that forces the bacteria into hiding in the first place, right?
Right. The stomach environment becomes incredibly hostile, full of this oxidative stress, so the bacteria shuts down to protect itself.
Think of ROS as a kind of cellular smoke that tells the bacteria it's under attack. And vitamin C is a potent antioxidant.
So by flooding the localized gastric environment with vitamin C, you clear the oxidative stress, you blow the smoke away.
That signals to the bacteria that the coast is clear. It fires up its metabolic engine, drops its Coccoid defenses, and turns back into a vulnerable spiral shape.
And that is when the standard antibiotic can strike effectively. That framing completely solves the contradiction of the clinical trials.
It really does. The trials that failed applied the treatment universally, likely at the wrong time, without understanding that vitamin C is merely the key to unlocking the door for the actual antibiotic to do its job.
Yes, but the author can't just leave it at the theoretical mechanism. If they want this guide to be truly authoritative, that conceptual slide must be immediately followed by a strict practical prescribing protocol.
So what would a concrete clinical application look like for this? We don't want to leave them guessing on dosages.
We'll follow this with a specific proposed regimen slide. For example, recommending a 500 milligram daily dose specifically alongside bismuth-based therapies for patients with a documented history of recurrent positives.
You narrowing the scope so the clinician knows exactly who this is for.
Exactly. Clearly label this as an adjunctive, off-label strategy aimed at ROS clearance to prevent the bacteria from remaining in the persistent state.
By giving clinicians a targeted, conditional protocol, you turn a confusing pile of conflicting meta-analyses into a sharp, actionable clinical tool. You empower the clinician to use the biology against the bacteria.
To recap, this critique highlighted 3 key areas for elevating the impact of the material. First, inverting the narrative to lead with the clinical mystery of recurrence before diving into the molecular biology.
Second, establishing a tiered diagnostic triage pathway to ground advanced tools like PMA-qPCR in clinical reality.
And third, framing vitamin C as a strategic Trojan horse waking agent with a clear, specific prescribing protocol.
Because when you're dealing with an infection that can literally unplug the machine and hide under the floorboards, you can't just keep swinging the mallet blindly.
No, you have to know exactly how to draw the mole back out into the light. We invite the listener to implement these structural changes and submit their revised presentation back in for further feedback.